Okay, unknown number 5, we're going to be talking about the gram-positive rods, and you'll get a number of these organisms, Rhodococcus equi, Truparella, Erysipelathrix, Corynebacterium, Listeria, and Bacillus. These are what we refer to as fastidious organisms, many of them. They grow a little slower than your other organisms that you're working with. They tend to be somewhat pleomorphic as well, meaning they're going to have different shapes to the cells. So it's really important for this lab to look at the gram stains, to look at your cell morphology, and then also to look at your growth on your blood plate to see the colony morphology. So we'll have photographs of all that on your Moodle site that you can take a look at. Each group will receive 4 of these cultures that I mentioned. So you won't get all 6, you'll just get 4 of the 6. And then again, the gram stain should be performed. That's going to be really important with this group. So you'll be, in this exercise with each organism, you'll be streaking a blood plate, streaking for isolation to look at the colonies. You'll be inoculating the TSI, and if you remember, you streak the slant and stab down into the TSI medium. You'll get glucose phenol red this time, so it's basic phenol red broth. It just has glucose in it this time. And again, if it turns yellow, then it indicates it's producing acid from glucose. Urea is an auger that, or the Urea Slant is going to be a test for urease production. You will streak the slant only, and if you get a bright pink coloration on the slant within 1 hour, that's indicative of a very fast reaction of urease production. But we'll also look at it in 24 hours to see if the whole tube has turned pink. So urease is really an important test with this group as well. Another medium that you'll be introduced to is Motility S auger. This is a medium that actually has an indicator in it that will turn red when the bacteria grow. But again, you'll inoculate this with a needle. You're going to heat the needle in the back incinerator to sterilize it, let it cool. You'll pick up an inoculum from the tube of your organism, and then you're going to stab that right straight down through the middle of the medium, not touching the bottom, and then pick it straight back out. If the organism is motile, it will spread out from that stab line, and it'll produce a red color that'll help you to see that. But if it stays right on the stab line, then that tells you that it's non-motile. So we'll be giving you demonstrations of that in a separate video. And you'll also get photographs of the reactions on your Moodle site.
Okay, using a sterile applicator stick, I'm going to take a colony off my blood agar plate, go into the Urea Slant, and streak the top, just like we did for the bile esculin. Okay, this is how we streak the citrate. It's going to be just like the bile esculin and the urea. We sterilize our wand. We take a little bit and we just streak the surface of the slant. Then we incubate that for 24 hours at 37 degrees Celsius. For the motility, we're going to do just a direct stab. This media is not quite as firm as all the others, like the TSI and the urea. It's kind of like you half made Jello a little bit. It's very squishy. So you just go one streak down, not all the way down to the bottom of the tube, but enough. And then we incubate that for 24 hours.